Well, I want to welcome everybody to today's lecture by Dr. Paul Wills. Most of you know me. I'm Dennis Hanasak, a research professor here at Harvard Branch and host of the series. So welcome to 2021. I think we're happy that 2020 is behind us. For this lecture series, today actually kicks off our 23rd year that we've been doing this. And our goal is always to give you a good taste of the research that we do here at Harvard Branch and that of some of the other experts in our fields who sometimes join us as guests. We look forward to resuming lectures to our audience here at Harvard Branch after the pandemic subsides. But for now, we're connecting with you virtually. And so far, it's worked out pretty well with our previous two talks. And I think many of us now spend a lot of time looking at our screens. And so this winter, instead of doing them as we traditionally do every week, we're going to do them every other week and alternate and possibly go longer into the year in that format because we're able to reach so many more people this way. So I just want to mention then that in 2 weeks, so not next week, but 2 weeks, on Wednesday the 27th, our speaker will be Dr. Esther Guzman. And her talk is the Deep Sea's Treasure Trove, Marine Natural Products as Potential Cancer Therapeutics. Esther is an immunologist. She works in our Marine Natural Products program. And her lecture will focus on the cancer biology side of Harvard Branch Drug Discovery Program, how it works, highlighting marine natural product compounds with exciting effects against cancer cells. So now if you're a regular attendee of the series, and I could tell just by doing a quick scan of the participant list that a lot of our lecture series friends are out there, you probably recall that in that group, the biomedical group, we have 3 scientists who for many years have rotated and given us a different talk every year on drug discovery. So this is your chance to catch Esther next time. She only speaks every 3 years. So she gets a great turnout. A lot of people are interested in cancer therapies and also just the great work that the whole group does in marine natural products. So hope to see you all in 2 weeks. Now it's a real pleasure to introduce today's speaker, Dr. Paul Wills. Dr. Wills is a research professor and is also our associate director for research here at Harvard Branch. He received his Ph.D. from Southern Illinois University at Carbondale. Paul has been here at Harvard Branch since 2006 and is a key faculty member in our Aquaculture and Stock Enhancement program. His main research interest is the production of marine fish for sustainable commercial aquaculture and stock enhancement in recirculating aquaculture systems. I could tell you a lot more about some of Paul's accomplishments in the last 14 years. That is the focus of his talk this year, though, so I'm not going to take any more that might take away from his talk. He's going to give us a very nice overview of that. I will just add that Paul is a wonderful colleague, and we are happy that he's here. We're even more happy that he can give this talk today. So please join me in welcoming Dr. Paul Wills for his lecture on fish culture research at HBOI, the last 10 years and the future. Paul, it's great to see you. And I should explain that Paul is doing this remotely from home. In this room, there's just 2 of us, but we have requirements here to be fully masked. And Paul did a little traveling, and one of our requirements is he has to spend a little time at home. And so this is a different approach, but we're going to try it. He normally doesn't dress this nice, Paul. You did a really nice job dressing at home.
My wife dressed me.
Oh, very good. Thank you, Paul.
Thank you very much for the wonderful introduction, Dennis. And thank you to everybody that is attending virtually today. So as Dennis said, I'm going to be speaking about what I've been doing at Harvard since I came to Harvard Branch. I'm going to go ahead and share my screen so you can see my slides. So you see my title is a little bit misleading. I say the last 10 years and the future, but actually I've actually changed it to the last 10 plus years because there was some key studies that I participated as a colleague in when I first got here that I wanted to highlight as well. And as I go through, I'm going to briefly talk about many different things that I've done since I've been here. Harbor Branch has afforded me a wonderful opportunity to do a vast array of different types of research related to fish culture. Now, when I'm talking about fish culture, many of you have probably seen me speak in the past, but for those who haven't, when I'm talking about fish culture, I'm talking about fish farming, and specifically my area, or we also call it aquaculture. My area within aquaculture is primarily with finfish, that being fish that have fins, like the goldfish you might get at the store or whatever. And so because there's this vast array of different work that I've been able to participate in, I've worked with a lot of different wonderful people here at Harbor Branch and in other institutes and other agencies. So now the work that I'm going to show you is all collaborative work that I've done. It's been a wonderful experience for me, and I hope to share that with you tonight. So to get started, once again, just to bring people up to speed that aren't familiar with aquaculture, I'll do a little bit of overview of fish farming and the importance of fish farming in the future for food production and for stock enhancement. So we think about fish farming as a way to produce food, but it can also be used for stock enhancement, and it's been widely used for this in freshwater systems especially. So why would we want to culture fish? Well, one of the primary things we think about with food fish is that they're a very healthy, low-fat protein source. Typically fish, especially marine fish, are very high in omega-3 fatty acids. Some of the freshwater fish aren't so high. Luckily, though, in aquaculture, we can actually adjust the types of fat that they're getting in their diets and boost the amount of omega-3 fatty acids. One of my colleagues, Marty Ritchie, always tells me when it comes to the oils in our fish, the fats in our fish, they are what they eat. So we can take advantage of that in aquaculture and ensure that the product that is being put out is high in omega-3s. Omega-3 fatty acids are very important, many of you know, and I've borrowed this slide from Marty because they reduce coronary heart disease, lower heart attacks, reduce cholesterol, lower blood pressure, and reduce inflammation. So omega-3 fatty acids are good, and seafood, fish especially, are a very good source of those. When we look at protein production overall in farming, fish farming has been shown to be one of the less resource intensive production systems relative to other animal proteins, so beef and pork, etc. So here the arrow highlights are showing you exactly where the fish are on this. This is a graph that was produced by a group called World Resources Institute that compared different protein production systems based on water use, land use, etc. And you can see fish farming are just above many of our plant production systems. Now, water use is a little bit higher than eggs, protein, and dairy, but lower than pork and beef. So overall, it does rank as very less resource intensive than those other meat production systems that are typical. The other thing that we want to take into account for why we want to do fish culture is our world's oceans. the fisheries in our oceans are basically at their production level. But we have an increasing demand for fish overall in our culture because it is so healthy. And you're seeing a growth, not only a growth of our population, which stimulates demand, but also a overall growth in the demand within that growing population. Today, approximately 50% of the seafood we eat worldwide comes from aquaculture. This is a graph from FAO, and it shows the relationships between fisheries and aquaculture production. And you can see this line here is our aquaculture, I assume you can see my arrow. This line here is the aquaculture production for human consumption. The darker blue line is fisheries production for food fish. The yellow line above that is fisheries production for all fish. So that includes a group of fish that we call industrial fish, which are used for oils and fish meals and such. But you can see here in about 2014 that we saw the crossing of the line between aquaculture production and other capture fisheries for human consumption, which is the point where aquaculture began producing more than 50% of our fish. And then for total fisheries, you can see the dotted red line is the cross, which happened back in about just mid 2019. Less than 1% of this production is coming from the United States from aquaculture. The rest is worldwide, with the bulk of it coming from China and Indochina. Then when we look at our fisheries overall, production in our fisheries plateaued. So the previous graph that showed the lines of fisheries production, we're not going to see a further increase in production. As you can see on this graph, another graph from FAO, we have an overall increase of fisheries over time from the 1950s to the mid 80s. What's interesting here is back in the 1970s, another faculty member who was at the time at MIT or at Woods Hole predicted based on ocean production, and was also a former faculty here at Harbor Branch, based on primary productivity of the ocean and the increase in trajectory of the fisheries, that the fisheries would plateau sometime in the late 80s. As it turned out, he was correct in his model prediction. And in the late 80s, we saw a plateauing of the capture fisheries, the total production. So the capacity of the ocean to produce more fish to fill the demand is not there. You can play around with the numbers others have, and you can increase this maybe by 10 or 12% at most, is what the modelers say. I'm not a modeler. I have to believe the facts that they give me. But you can see at the time that break point occurred, that's also when aquaculture development worldwide started really increasing, and you saw this uptick of aquaculture production to fill the gap. So oftentimes people think that aquaculture competes with capture fisheries, where in fact it does not. Aquaculture fills a gap in demand that's for fisheries product worldwide. So in total for the US, this amount of production, or this lack of production, is related to the fact that over 90% of the seafood that we consume in the US is imported, resulting in a trade deficit of about $14.9 to $16 billion in the US economy. From year to year it changes, but it fluctuates between about 14 and 16, and I've seen figures upwards of 17. If we look at our oceans on this concept of whether we can produce more, also, if we look at our ocean fisheries, about 77% of our oceans fisheries are fully exploited, overexploited, depleted, or recovering. So once again, relative lack of room for increasing the amount of fish that are produced. A picture of what our markets look like for aquaculture products in the U.S. In 2018, it should be about the same coming up when we see figures for 2020. U.S. consumers were eating about 16.1 pounds per person of seafood. Now, some people obviously eat more and some people eat less, but you get an average of about 16.1 pounds. This is the list of the top 10 species that are consumed. The ones that are in light blue are aquaculture species, or species that are primarily coming from aquaculture. Of course, salmon comes from both aquaculture and wild, but 2/3 of that production is actually coming from aquaculture. If we get rid of the non-aquaculture species, then you can see these are your aquaculture species. And then among those, these are the finfish that are being produced. So that kind of gives you a quick overview of the aquaculture industry worldwide and US and consumer demand. So you can kind of get a sense that aquaculture of fish and other species, and I say other species because not only are we seeing culture of fish, but we see culture of mollusks and seaweeds and all sorts of other products now, and all of those are important in producing the foods that we're going to need into the future worldwide. One of the things I like to do for my students is I like to show them where aquaculture was when I started my career. It's kind of interesting exercise to do, because when we think about aquaculture, it's still relatively small industry. But this graph only goes to 2013, so these lines continue to go up. But when I was born, aquaculture, if we think about it being small now, when I was born, aquaculture was very small, almost non-existent industry. Even when I went to college in this period of time and graduated, aquaculture was still relatively small. And oftentimes I would talk to people, they would ask me, why are you getting into that? I guess I was a little bit of a prestidigitator. I guess I was really seeing the future in this industry. To me, it was very exciting. So by the time I got my PhD, we can see there was a massive increase. And that increase is still going up, and it ought to continue to go up over as time comes by. So after I finished my degrees, I did postdoc work, and then I took a job with a commercial aquaculture outfit in Illinois, and you can see that's me working in a pond. So I was managing the farm, and I learned a lot in book school, you might say, but to get a really good appreciation of what the industry needed and that sort of thing, working on the farm really helped me do that. One of the things I'm really impressed about with all the aquaculture faculty and scientists and staff at Harbor Branch is that almost every one of them has experience in the industry, working on farms of one sort or another. So we, as a collaborative group, we really have a wonderful insight into the needs of farmers. We have contacts with the industry that is very helpful to us. So this allows us in our program to really address the sorts of questions that farmers need, which I find to be extremely unique in the research world. So after I left the farm, I was invited to come to Harbor Branch and work. And of course, the owner of the farm was not happy, but he's still a good friend of mine. We visit every Christmas. when I go up to Illinois. That's where my wife's family is from, so we spend Christmas there, which is why I'm at home right now. So, but I came to Harbor Branch to work on a particular project, which is a collaborative project with USDA ARS, developing inland recirculating systems, especially tailored towards low salinity culture of marine finfish. So my primary work up to that point had been with freshwater fish and freshwater culture systems, which I think dovetailed well with the engineering and husbandry and biological work that the group was doing related to developing these low salinity inland recirculating systems. It was a wonderful group of USDA scientists and Harbor Branch scientists and staff that were working on that project. Overall, though, that project gave me a kind of in my career and the way I approached collaborations, a kind of a blueprint for how to proceed with my, continue with my career. And so I have kind of a bunch of different areas of interest that I kind of work in, and where my collaborations have developed. One of the first, I'll say primary, is finfish species development. So myself and all the other scientists at Harbor Branch, we do quite a lot of this species development, not only for finfish, but the other groups as well, that being clams and mollusks and other invertebrates, as well as plants, etc. So Dennis and I collaborate quite a lot on farming of aquatic plants, be that seaweed, or we did some work with freshwater plants, etc. So within the finfish species development work, quite a bit of work being done with food fish and stock enhancement species. One of the things with food fish species is, since we're working with collaboratively with groups all over the country, and we hosted a workshop a few years back and worked with each of these collaborators and developed a list of species of primary interest currently in the U.S. This is not an all-encompassing list. There are many other species that people are interested in, but this gave us a path forward for looking at various species. Once again, the species in light blue on this table are species that we've worked with here at Harbor Branch, and then other species around the country are working with the ones in purple. And we have interest in some of those species. Like currently, we've got a collaboration with folks that are working with Almaco jack and some of these other species. So these are the food fish that we're interested in, have worked with at Harbor Branch over the years. On that USDA project, one of the primary species we worked with when I was on the project, prior to me coming here, they worked with black sea bass and flounder and some striped bass and some others. But when I came onto the project, we were primarily working with Florida pompano, and then later did some work with cobia. Florida pompano is a very nice fish. It's if you eat fish and you like fish, Florida pompano is probably one of your favorites. It's listed as one of the best eating fish of all fish in the ocean by many people. It has very high market value. A lot of that is attributed to the seasonality of the product and the relative scarcity in the commercial fisheries. In aquaculture, it's nice that it will accept a prepared diet, so we can feed it a pelleted diet, which we can control the ingredients of. It is very adapted to intensive culture systems like we work with, has pretty high initial growth rates, and there's a pretty good research base which is growing for how to culture them. Their primary area of natural distribution is in east coast of the United States and Gulf of Mexico and the east coast down into South America. They're a shoreline fish. They are primarily in the surf zone. They support a good sport fishery, and at market they have very high value. I've seen, this is $18.95 a pound for fillets. I've seen fillets upwards of $22 a pound at some points. So we did quite a lot of work. I'm going to show you the results of one of our projects that we worked on, which was looking at the relative productivity based on density. So most of these projects we use techniques that are used in production aquaculture, commercial production aquaculture. We try to do them at a scale that is relevant. Of course, we can't do it at commercial scale because that would be very large tanks. But we try to do a larger scale than most research outfits, and so that the results are relevant to production aquaculture farms. So we looked at density. An interesting thing we found is that the low densities and high densities are about 30 kilos per cubic meter versus 15 kilos per cubic meter. So we actually saw a density effect with pompano, where if we increase the density even more, the productivity reduced. So of course, this is a concern, but it also points to the densities that a farm would have to work at in order to effectively produce pompano. Another thing that we found that was very interesting with this, that came out of this study, is we actually segregated males and females and looked at the relative size of the males and females. The females were actually a little bit larger than the males, which is interesting. This was seen in wild fish, but in the wild fish, you've got multiple ages and that sort of thing. It's difficult to actually precisely separate out the different age classes. Here we have a whole group that's all one age, and we're seeing that the females were a bit larger than the males. So there are things that we can do to do all female culture. And you probably saw in my bio that this is an area that I used to work in before I came to Harbor Branch. So someday I hope to revive this and possibly look at doing this with pompano. Another species that we worked with on that project was cobia. This is actually funded by both USDA and Florida, or the National Sea Grant Program. We were looking at culture of cobia in recirculating aquaculture systems. Cobia is a really wonderful fish. They support sport fisheries. They produce a very large fillet. You can see this fillet here. That's about almost a 30-inch tray that's in. So extremely large fillet. They're pretty cosmopolitan around the world, but they don't exist on the west coast of the U.S. Interestingly, even though they're pretty much everywhere else in the world. They grow very fast. So this is the same group of fish. So this little guy is a sibling to this guy here, but this is 8 months later. So extremely fast growth with this fish, very high food conversion efficiency. It's a wonderful fish to work with. So we did several different studies looking at different types of feed, and then we also repeated a density experiment similar to what we did with the pompano. This is the feeds that the fish got during that experiment. And I just wanted to point out that with pompano, when we get to market size, this 5 millimeter size is about the maximum size they get, somewhere between 5 and 7 millimeter. These cobia we were given a 12 millimeter size, and they could have taken something even bigger than that. So looking at the density results, a nice result that we found was, unlike the pompano, at the 30 kilos per cubic meter density, this is 30, 25, and 15 cubic meters per density of it's kilos per cubic meter of water for density in the tanks. So at the higher density, the fish were not showing reduced growth like we saw with pompano. So growers of cobia and recirculating systems could go at higher densities. And that would be something we would want to test going into the future. Here's some pictures of the product, or of that density in the upper left-hand corner. You can see a tank full of cobia at that high density, and you can tell from this picture that they are really packed into that tank. There's a lot of fish in that tank. The water's really nice and clear because the recirculating systems work very well, and it produces a real nice product for consumer. Well, we don't actually sell product, obviously, we're doing research. The nice thing about it, though, and the nice thing about being an aquaculture researcher is when you're done with an experiment, you get to eat your experiments. So that's always a nice thing. Lately, we've been doing, I've got some funded work from Florida Sea Grant looking at pompano again, actually looking at some new technology for feeds, types of feeds and feed ingredients, investigating probiotics. You're probably all familiar with probiotics, that you can take probiotic pills to help your digestion. Same thing works with fish. You can have those probiotics in their diet to help them improve digestion and also reduce diseases and that sort of thing. So we've completed several studies already on this project, one looking at the probiotics themselves in juvenile culture, which are these pictures on the left. So we grow them, and then we take a lot of data on their blood parameters related to health. You're familiar with CBCs on animals, differential blood cell counts, and other measurements of health. And then another experiment we finished is looking at a new type of pellet that's actually soft. One of the things we find with pompano is related to their wild feeding behavior. The pompano actually will grab a pellet and crunch it up before it swallows. This is similar to what they do in the wild. They're eating crustaceans, crabs, and that sort of thing. And in the wild, they will grab those crabs and crunch them up and sift out any sand and legs and that sort of thing that they don't want to eat and consume the rest. Well, we found that they were also doing that with the pellets that we fed them. So we'd take and grab that pellet, and then they would crunch that pellet up, and then they would sift the fines, the fine particles of that crushed pellet through their gills and lose it. And we found that they were losing upwards of about somewhere between 15 and 18% of the diet. Never got consumed. So if it ever gets into the belly, it can't then be turned into fish flesh. So the soft pellet is a new type of diet that we're testing that we hope is going to solve that problem, since when they grab the pellet, they'll crunch down on it. Instead of fracturing, it'll just squish down like bubble gum, and then they'll just swallow it. So once again, we're also looking at probiotics in the soft pellet. So some of the fish got pellets that had probiotics, some without, and we look at the health of those animals using the various different health-related parameters as well. We just recently finished another experiment looking at pellets. So this time, instead of, this is once again related to looking at how they consume that pellet and reducing the amount of crunching of that pellet. So, but rather than using a special soft pellet, we're looking at changing the pellet size they get at different sizes during production. So actually reducing, how does the reduction in size of the pellet affect the total amount of wastage? So my graduate student, Tyler, there, this is part of his thesis work as well. And he was going through and measuring the waste coming out of the tanks periodically during the study. Did a wonderful job doing that. And then they got fed from smaller fish all the way up to harvest size. And you see Zach here on the right taking measurements and Molly writing down his measurements. So alongside this, throughout the entire production, they were measuring gape width, and Tyler will be using those different parameters in order to assess how the fish did in total using the different pellet sizes over the production cycle. And then looking at the effects on dress out and other parameters of flesh quality. So that's kind of a look at the types of things we do with food fish production, aquaculture. We also do a lot of work with juveniles and in the hatchery with how to spawn and rearing the larval fish, pretty much taking an entire life cycle approach to the culture. And this is once again something that came out of the work that the collaborative work we were doing with USDA. It's kind of a philosophy of the lab as a whole and the way we work. I haven't shown you a lot of that because I really have a limited amount of time, and I want to show you a lot of other things that we do. And one of those is stock enhancement. Within the realm of stock enhancement, so this is production of fish for stocking, but it also represents investigating the reproductive biology of fish. So you can do stock enhancement directly by stocking, or you can do stock enhancement by having a better understanding of the reproductive biology of fish and then protecting or managing the resource in the wild rather than actually stocking. So when I'm talking about stock enhancement research, that I'm talking about it in those two different contexts. So in those contexts, we've done quite a bit of work for the Florida Wildlife Commission looking at redfish production in recirculating systems. We did this back in the 2008, 2009. So prior to that, most redfish are being produced to very small sizes in ponds and then being immediately stocked. There was some work that the FWC had done looking at different stocking of different sizes. They called it Project Tampa Bay. You can go on the web and find that, and it's a very interesting study. And they looked at stocking of different sizes of redfish at different times of the year and that sort of thing. So our involvement in that was to look at efficiency of production of redfish up to the various different sizes in the context of a recirculating hatchery. So you can see Richard here is grading redfish early on in their life. They can be a bit cannibalistic, so grading is required in order to reduce cannibalism in the culture practices. And from that, we were able to develop some information for the state on how well they do, how healthy the fish are coming out of recirculating systems, what the relative cost of different sizes of fish was, and that sort of thing, which are inputs that the state, for instance, would need if they're managing a hatchery, for instance. Currently, we have a project with Bonefish and Tarpon Trust that's funded by Bonefish and Tarpon Trust through funds, their funds, and matched with funds from the National Fish and Wildlife Foundation. This is our bonefish reproduction research project. And like I said, this project isn't geared towards producing fish for stocking, but rather using aquaculture techniques to investigate the reproductive biology of bonefish that can then be interpreted to wild fish. In this study, we look at both wild populations and we look at populations that we have in the lab. So we have a clear picture of what's happening in the reproductive biology in both situations, and how and one helps us interpret the other in essence. So in this project, I'm working with, I have several mini collaborators. This is John Schenker in the picture next to me. We're looking at a broodstock bonefish. And then you see the picture of a little larval bonefish that we produced as part of the project. So the reproductive, but these larval stages of bonefish are very difficult to find because they're out in the open ocean, and it's logistically difficult to find them. And when, you know, they get destroyed when you're in the sampling gear. So a look at a larval bonefish this age is only really possible using these types of techniques. When we started the project, this is kind of what we knew about the life cycle, which is almost nothing. We knew where broodstock were, we knew what the eggs looked like, but we didn't know what eggs that were mature and ready to spawn looked like. This project has been going on for just under 4 years now, and this is where we are now. We've done a considerable amount in filling in the various different gaps within the life cycle. We still have some small gaps here and there, but we'll be addressing these in the next year of the project. And I believe we'll have a very good handle and have filled in the entire list of questions that we have related to reproductive biology of this fish. But we have seen now all the different life stages of developing eggs, all the life stages of larvae up to about 96 days. And once we still don't know feeding, we've attempted several different types of feeds. We have ideas of things to try when we get our next spawn of bonefish. And we have incidentally been able to manipulate the broodfish with light cycles and temperature and that sort of, and induce them to spawn in the laboratory and produce larvae. So that was a huge step for us in accelerating to fill this particular gap. And I'm confident we will. That's part of the future work for this project, and we'll get there. I'm certain of it. So I'm going to step away from that and talk now about aquaculture system technology and develop for land-based systems, especially, which is kind of the area that we at Harbor Branch excel in. And this is the area, one of the primary areas of that initial ARS, USDA ARS project that I came in on. I had been doing some system development prior to this in my previous life before coming to Harbor Branch, but this was really accelerated in my career in coming here with such wonderful colleagues that were working in system in technology development. So one of the primary types of systems we work in are what we call recirculating aquaculture systems. These are really nice systems. They allow year-round growth, a lot of improved efficiency in water use, et cetera. They are a bit power intensive, but these are some of the questions that we're addressing. They enhance biosecurity because you have complete control of what goes in and out of the farm. You have increased food safety, containment of escapees. So no possibility of animals escaping and affecting natural populations. So from an environmental sustainability perspective, relative to some other aquaculture systems, are very sustainable. For those of you that don't know how a recirculating aquaculture system works, it's what we call serial reuse of the water. So the water that's in it, so this represents a tank of fish. The water that's in that tank flows. We feed the fish obviously eat. They pee and poop in the water because that's what fish do. We want to control that from becoming toxic. We run that through various different filtration systems, solids filters that remove the solids from the water, the solid waste, primarily the feces. We can concentrate that sludge and remove it. Then the water that's had the solids removed will go into biofiltration. That biofiltration will remove the ammonia and nitrates, the ammonia and nitrite, from the water, reducing it to relatively non-toxic nitrates. And then the water can be returned to the system. Now we can do some reoxygenation and that sort of thing to the water. before the fish get it. And that way, everything in the culture tank is going to be healthy, and they're continuously having their water replenished and refiltered so that there's a nice, clean environment and healthy environment for the fish to grow in. We do lose some water through evaporation, but we can make up that water. We have complete control over the amount of water, but the amount of makeup water is reduced by improved technology. So that's what the low salinity culture system was related to. In the context of that ARS project, we were developing systems for nutrition research, larva culture systems that were all recirculating. Quarantine systems. Quarantine is often used for animals coming onto the farm, or if you have something that gets diseased, you want to quarantine them. So we have specialized systems we develop for that. Systems for grow out production. I've shown you some pictures of that already. So large scale production systems. And then after that, Dennis, I actually used the knowledge from that to help Dennis in developing a seagrass nursery. Now, this isn't fish, but kind of throwing in a little bit of the plant and how the technology development. This was a project that was funded originally by Harbor Branch Oceanographic Institute Foundation. And Dennis and his group is now growing seagrass to beat the band with that recirculating system. So the technology is applicable not only to fish, but also to plant production. After that, I got some funding from the state through the Aquaculture Review Council to do a large scale demonstration system. This is a panoramic view. So this tank, you can see the tank here on the left and right. That's the same tank because it's a panoramic, but you can see all the filtration systems. So essentially a scale up of the other systems that we had developed on the USDA ARS project. We produced a crop of about 7,000 pounds of pompano in this tank. And incidentally, we actually donated, that's way too much for me to eat. So we actually donated that to a local food bank who was very much appreciative of that. The systems like this also allow us to look at efficiencies and and compare efficiencies of solid removal and that sort of thing for our smaller research systems. Other things, technology that we developed on those various projects were control systems for oxygen monitoring and feeding, linking to things. They're internet accessible, et cetera. So technology improvements for farm management. One of the issues with recirculating aquaculture is off flavor. You can get off flavor as a result of the various filtration. So I had some funding, once again, from Aquaculture Review Council of the Division of Aquaculture, or Division of Aquaculture for the Florida Department of Ag and Consumer Services. And I was able to look at multiple different types of systems for removing off flavor compounds from the water. So what happens is the bacteria and that sort of thing in the water will produce these compounds, primarily methyl isoboronyl, which produces a musty flavor, or geosmin, which produces an earthy flavor. And so I was looking at technology for how we could get that out of the water, because if it's in the water, the fish will absorb it, and it's not toxic. They're non-toxic, but they actually give the fish an off flavor, a musty or earthy off flavor, which is nice in mushrooms, but is not good in fish. So we tested several different devices that we developed here at Harbor Branch, hydrophobic media filters, which use basically paraffin, ozone contactors, and then also a titanium dioxide photocatalytic degradation reactor, which actually uses plates that are bombarded with ultraviolet light. It's essentially the germicidal lights that you see in hospitals and whatnot, that light blue light. The plates themselves are coated with titanium dioxide. It's crystal clear, so you can't actually see that the plates are coated. But there's a reaction that takes place at the surface that will break down these various compounds. And once they're broken down, the fish won't absorb them, and the fish won't then be off flavor once they're harvested from the recirculating system. So the extension of that standard type of recirculating system for us at Harbor Branch was this new type of aquaculture system called an integrated multi-trophic system. And this, we were working on this because we had some issues with our recirculating systems producing waste. So the recirculating system it converts the waste to less toxic form, and then it has to either be filtered or discharged. But if you're discharging something, you're producing a waste. And we wanted to eliminate the waste. How the approach that we took to doing that was adopting integrated multi-trophic aquaculture into our research agenda. Integrated multi-trophic aquaculture will turn those wastes, so we stop thinking about waste as waste and we start to think about them as resources and how we can use them as resources. The way we do that is a much more complex system, but that system allows us to produce multiple crops with each of the various crops using a quote unquote waste as a resource to produce that secondary crop. So the fish are cultured, they're eating feed, they're producing waste. The waste goes to a specialized type of filtration system that we've extended from the previous recirculating work that we did with USDA ARS. And then now we're using the nitrogenous waste, the nitrates and ammonia that's produced in order to grow plants or grow. The solid wastes are being used to grow other animals or whatnot. And so now you've got multiple crops coming out of a single system, and you're being essentially more efficiently using prepared feeds that are going to the fed culture animals, be that fish or shrimp. Plants are one. So I wanted to show you that I actually do work with plants as well as Finfish. So this is some work that Megan Davis is heading along with Richard Baptiste. And we were harvesting an experiment that we finished during the pandemic. So you see everybody's masked up and we're harvesting the plants that were produced in this system along with the fish, etc. Other things that we do, we produce other products in the system, seaweeds and whatnot, biofloc, which is the solids. So rather than using the solids directly, we convert them into biofloc, and then we can make feeds out of these ingredients. And we've tested how they work in growing other species, for instance, here, red drum. So these biofloc and seaweed are incorporated into the pellets that we feed the fish and see how well they do. And what that's doing is substituting for the fish meal that would otherwise be in those diets. With our engineering group, we are doing a wonderful project looking at using drones for pond-based aquaculture. So rather than having personnel working out on the ponds in the middle of the night, this is a picture during the day, but this is generally something that's happening at night. They're driving around the ponds, taking oxygen readings to ensure that the animals are going to be safe throughout the night, that the oxygen in the ponds isn't going down to the point where the animals wouldn't be safe. What we're working on is a system now that will automate this with autonomous drones, basically a swarm of drones that will fly around the farm, taking water samples with. So this drone would have an extendable probe that went down into the pond, took the oxygen, transmitted that then to base stations, and there would be artificial intelligence prediction models that would tell the operators then which ponds are likely to have dissolved oxygen problems, so that the operators can then move aerators and whatnot into place into those ponds immediately and help to reduce the overall cost of operation, because you're not having to aerate. You're aerating more efficiently in essence. Here's a picture of our prototype drone that we've flown for that system. Another important area for us in total, because we are doing things that we want to go to farms immediately, is our education technology transfer. We do that through producer workshops. We hold these workshops. Oftentimes they're related to specific projects. This is related to that commercial. This is pictures of a workshop related to that commercial demonstration project with Pompano. We do that through our Indian River State College aquaculture curriculum that many of our professors here teach. Traditionally, I've taught water quality and finfish aquaculture. We have our students, they're learning hands-on the techniques, et cetera. Of course, now with COVID, it's mostly virtual, but the instructors that are doing that, or the professors that are doing that, are doing the very best they can to give as good an experience to those students virtually as possible. We have stakeholder workshops. We work with multiple stakeholders, that being agencies and farmers, et cetera. That list of fish I showed you at the beginning came out of a series of workshops we've been doing on the status of marine finfish for U.S. aquaculture over the last couple of years. And then we have our research and education partnerships. So we partner with a lot of different groups. Many of these are research related, many of these are education related. We also have industry people, various different farms, for instance, that we work with. And the idea there is if we're doing research on farms with farms, then the technology is immediately going to where it's needed, rather than sitting in a journal somewhere that the farmers don't necessarily get access to. So this is an extremely important part of our overall research approach, is these partnerships. Current and future research, I need to finish up here. I have these 4 projects, primary projects going on. I have a lot of other little projects as well, but these 4 projects are going on. We have a new collaborative research project that started last year with USDA ARS. This is a true collaborative research project. We will have USDA ARS scientists on site, just like we did with that original one that I came here to work on. This one is directed towards producing or improving enhancing production of warm water marine finfish seed stock. So these would be the small fish that need to be then stocked out for grow out on farms that would then go into the food fish market. We have the ongoing project that I showed you some of the work from Sea Grant with commercialization of Pompano. Our IMTA, Integrated Multi-Trophic Aquaculture project, funded by the Harbor Branch Oceanographic Institute Foundation, is ongoing. And then our research with Bonefish and Tarpon Trust on bonefish reproduction is also ongoing as well. Obviously, when we're doing projects, we always generate many questions, and we're constantly looking for funding sources to address those questions and help us to move the science of aquaculture forward for the industry. So the work we do is very much applied to, and the idea is to get it to the industry as quickly as possible. I promised earlier I wanted to give some acknowledgments to the many people that I've worked with over the years. I know that I've missed some people on the list. That's why I've got the ... But there have been so many people I've worked with as scientists and staff and researchers, et cetera. It's always been a pleasure. It's been a pleasure to work at Harbor Branch. This has been one of the most wonderful jobs that I've ever had since I started my career. Kind of fits the saying that Edwin Link said, the best job to have is a job that you enjoy. So I paraphrase that, of course. These aren't in any order. They're just kind of as I remembered them. So, you know, nobody on this list is more important than the others. They're all very important to me. And I've been really happy to be a part of the various teams and whatnot that have done all this work over the years. This is not my work. This is this group that you see on this slide. It's their work that I was lucky enough to get to participate in. So that's all I have for you. It's I know there's questions. I'm sure people have questions. Obviously, I couldn't cover the detail of 14 or so years of work at Harbor Branch, but I hope that I gave you kind of a good overview of the sorts of work that we do for aquaculture research and development. I hope you've enjoyed my talk. I can't wait till we're back giving talks directly, because I like to have the direct audience interaction, which we don't have. So.
Paul, thanks so much. And again, I'm sorry that we're not all together, because you would hear big clapping going on right now. So just to remind everybody, and we'll put a slide up so you can see the reminder how to do that. Down the bottom of your screen, you should be able to find a Q&A box that you can type in your questions. And I will work on that with Paul, help out on the moderating. If you don't see it right away, if you're not too familiar with Zoom, sometimes you just need to put your mouse or cursor down along the bottom right below what you're seeing of that slide, and you should get some information about muting and video, and somewhere in there it'll say Q&A, and you can just type your question in. So one that came in early on, Paul, that you can probably actually see if you can do that. Can you tell us, this is more about the fisheries question, so it's not so much an aquaculture question, but there is a comment about aquaculture here. Can you tell us about the latest status of turtle excluders as to how well they have been accepted by fishermen, for example? So maybe not everybody knows what that is. You might have to explain. And then there's a comment, if you want to address this too. We do love Harbor Branch's culturing of clownfish. So it's kind of 2 for 1.
All right. So I've not personally done any work with turtle excluder devices, or what for a while they started calling trawling efficiency devices, or TEDs. But when I was in undergraduate in Louisiana, this was a real hot and button topic with commercial shrimpers, especially because they felt like turtle excluder devices were, a burden. It basically was a change for them. So I don't know. I think that they're much better, more accepted by fishermen now than they were back then. But I really haven't, that's not really an area that I've continued to keep up with. Sorry, I can't give you a good answer to that. Now, as far as culturing of clownfish, with Harbor Branch, our work we really don't do any work with clownfish. The clownfish that are cultured on Harbor Branch property are actually cultured by a private company. Their farm is located within the boundaries of Harbor Branch, but on Harbor Branch Foundation property that is located within the boundaries of Harbor Branch Oceanographic Institute. That company is called Oceans, Reefs, and Aquariums. And they're right across the road. So we're very, very friendly and familiar with them. They ask us questions. We ask them questions. They're doing marine culture just like we are. So, if our rotifers are used to feed the larval fish, if our rotifers cultures have problems, we get rotifers from them and vice versa. So it's really, they're in the business to make money. We're in the business to do research, but it's, fundamentally what we do is very similar to what they do. So, and, it's nice to have them on the property. And the type of fish that they're producing, the clownfish and the other marine species that they're producing in land-based recirculating systems. So they're very sustainable as compared to the type of the ornamental industry that's out there collecting fish from the wild. A lot of these fish are Pacific fish. They come from Pacific. They would normally come from Pacific Islands, and the techniques that many of those fishermen use are very damaging to reefs. So the fish that Ocean Reefs and Aquarium produce are not are sustainable. They're not damaging the reefs. They're being produced in using the culture type, the sorts of culture systems that we use. So very noble effort by that group for certain.
Okay, here's a quick one for you, Paul. Someone wrote in and said, I'm not a scientist. I assumed all fish had fins. So what are finfish? I think that's a really good question. I think sometimes we make some assumptions, don't we?
Yes, so that is a very good question, and I always try to make the distinction because it depends on who you are and how you've been, where you've been brought up. So when we talk about fish and shellfish, so to distinguish shellfish from finfish, we often say finfish. All right. So many people think when they think of fish, they in general just think of seafood in general, which would encompass both finfish, shellfish, and such. So to make that distinction, we say finfish. That way, it's clear to people we're talking to that it's not shellfish. Not that shellfish are bad. Shellfish are great, and in our integrated multi-trophic system, we grow shellfish alongside of our finfish. So
this is right up your alley. You talked about fish pellets. What is the composition of fish pellets? If they contain fish, where does that fish come from? Again, another very good question.
Yeah, that's a very good question. So fish pellets are compounded out of multiple different ingredients, and they do contain fish. Those fish are in that group of fish that I early on called industrial fish. So these are species of fish that you generally wouldn't necessarily want to consume directly. So anchovies, menhaden, they're generally very bony and oily fish that most people really wouldn't consume directly. They're, to most palates, they're pretty off-putting, but they grow very quickly. They're, in the US at least, they're fished sustainably. They can be rendered into fish oil and into fish meal. Those fish meals are traditionally used in agriculture. So most of our other protein production systems do or have or still do use these proteins. Now, aquaculture now is one of the primary users of the fish meal and fish oils. But there are many other uses for these things. Very fine lubricating oils are made from these fish oils, and you see them also in supplements. But one of the key thrusts in research, and we participated, and I mentioned it with the redfish work that we did with products from the IMTA, is how do we reduce the total amount of fish meal that we have in the diets that we're feeding our fish. So ideally, we would want to eliminate it entirely. That's what the chicken industry has done. The chicken industry used to heavily rely on fish meal to produce chickens, but through research, they have been able to reduce substantially the amount of fish meal in the chicken's diet, while maintaining a good product, good flavor in the product, and maintaining good growth in the chickens. A lot of that's through nutrition and then also selective breeding. So selective breeding is another aspect of our new USDA ARS project that we'll be doing in conjunction with continuing our work with fish meal reduction in the diets. So we want to reduce as much as possible both the fish meal and the fish oil So we're reducing the burden of aquaculture on those marine fisheries. Like I said, the fish that are being used for fish meal are fished sustainably, and they're not going to be fish that you would want to eat directly anyway. So this is kind of a bioconversion to a more palatable diet. And then the other aspect in our research is improving the food, what we call food conversion efficiency. So oftentimes you might hear it as termed as the fish in, fish out ratio. And aquaculture as a whole has substantially improved its fish in, fish out ratio to the point where today the industry as a whole on a worldwide basis is about 1/2 pound of fish that goes into the diets produces a pound of fish. Now that number is probably 2 years old, so it's probably improved even since I saw that last number. So we're actually producing a net increase in protein available of fish in the aquaculture industry worldwide. Now specific fish are higher, but in the industry worldwide, we're actually producing net production of protein relative to the fish that's going in.
It's probably more than you
go ahead. Know, but I'll try to.
We got one from a former summer intern, Nolan Barrett. How are you? When do you believe that IMTAs will be more widely used, both in the fresh and marine aquaculture systems? They look very impressive, and I can't wait to see more of them.
The sooner the better. I think Dennis is one of the collaborative scientists on that project. He is responsible for the algae work. We have a wonderful team. I one time kind of did a quick calculation. Fully half of the scientists on site that are working day to day on site at Harbor Branch have, in one way or another, contributed to that integrated IMTA system and the knowledge that we now have about it. So the key is convincing farmers that it's technology they want to develop. And if you look worldwide, there's been an uptick. Oftentimes that uptick first appears in scientific literature, but there's been an uptick in, I'll call it chatter about IMTA systems worldwide. So you're seeing more and more adoption of the technology, and we're at the forefront of development of land-based recirculating integrated multi-trophic aquaculture, as opposed to it's also applied in open ocean systems.
Okay, we got several really good questions in the queue here, and so I'm going to say this is like your 5-minute warning, Paul. So if you want to more questions, you can answer them quicker. It's up to you. Ethan says, why is there a common perception that aquaculture fish aren't as healthy as wild fish? And is that really the case? Again, I think it's a really good question that a lot of people wonder about.
Myth and misperception is primarily there. So there was actually a talk that was done by Marty Ritchie and Megan Davis a few years back to address the myths of aquaculture. And there are multiple different reasons. And, once again, you follow the money, you'll see that there are groups that are opposed to aquaculture because they see aquaculture as competitors. Like I showed you in an early graph, aquaculture is filling a gap that the commercial fisheries can't fill. So aquaculture is not a competitor to the wild fisheries. The wild fisheries plus aquaculture form a whole of our seafood supply. So early on, though, aquaculture was really seen as a competitor. There were groups that really started to push this narrative. A lot of it is based on junk science or no science at all. So to answer, is this really the case? I'm going to say no. Aquaculture fish oftentimes are healthier than wild fish.
So this is from Red Jones. He's one of our regulars also, and he has a very thoughtful question. You brought this up, Paul. You talked about chicken, so we know you know about that. Chicken is much less expensive than fish. How much more quality of food and health is obtained by eating fish to make it worth paying the higher price over chicken?
So you're right, chicken is less expensive than fish, but we are where chickens were back in the 1950s. And there's a reason why a former president said he's going to put a chicken in every pot, because back then chicken was not inexpensive compared to other protein sources. So they've got a multi-decade jump on fish farming. And I would predict that in the next few decades, we'll see the actual cost of fish coming from aquaculture going much lower. A lot of the high prices are related to pricing for wild fish and the costs for production, et cetera. So better science to improve efficiencies, et cetera, will bring the prices down. And is it better? Is it healthier to eat? You know, that's for a nutritionist to answer.
Yeah.
Everything that I see says that yes, it is healthier to eat. But I like chicken. You know, we have to have a varied diet. We're humans, we're omnivores. We have to have a varied diet.
Yeah.
So, you know, educate yourself, make choices based on what you've seen. Pay attention to facts, pay attention to science rather than the myths that are out there. Personally, I would say eat more fish.
Yeah.
But
Sounds
I'm
like
biased.
it's commercial. So there's going to be, this will be the last question, but what I'll do is I'll weave a couple of them together, okay? But they all kind of address the environmental impact of aquaculture. So one of them is, what's the environmental impact of large commercial aquaculture farms? And then the other one is kind of a subset of that, or maybe more specific to some of the things you're doing. What are the downsides of aquaculture that you and your team work on besides reducing fish meal, fish oil, and waste? So both of them are getting at the issues of, you know, what are the challenges and maybe what are you doing about it?
So environmental impacts, there are multiple potential impacts. It all depends on the type of aquaculture farm you're talking about. So a large commercial farm that's pond-based is going to be very different from a large commercial farm that's recirculating based, which is going to be very different from a large commercial farm that is open ocean aquaculture. Each of them has its own subset of different potential impacts. And each of them has to address how to mitigate those impacts in order to operate in an environmentally sustainable way. Now, mitigating those impacts, for instance, in open ocean aquaculture may be more difficult than in recirculating culture. But recirculating culture has its issues as well. Recirculating culture is much more energy intensive. For instance, you have pumps and that sort of thing that have to keep the water moving. So you have that type of impact on the environment. Now, if you're able to use solar, wind, and that sort of power, then that impact can be mitigated. But we need to see development in those sectors in order to support a large commercial farm's use of those types of power. Now, many other industries are moving to those types of powers. Our energy systems in the U.S. are substantially moving to those types of power sources. So a lot of that is going to be self-correcting for land-based commercial aquaculture. The one thing I don't want anybody coming out of this to do is pigeonhole all of aquaculture based on one of those types of aquaculture. It's very easy to do. That's oftentimes how we end up with myths and that sort of thing. An individual will say aquaculture is not environmentally sustainable, and they're thinking about one certain type of aquaculture and making a broad brushstroke across all aquaculture systems. So you have to look at each type individually, assess the types of environmental impacts, how they can be mitigated, and then go forward in making an assessment of whether or not those impacts are sustainable or not. You know, ponds are essentially can be operated as a closed system, so very low environmental impact from a waste perspective. But the total production per unit area is lower than, say, recirculating or cage base. So they do have an impact in their need for larger tracts of land in order to be successful. So, you know, every type of system has its trade-off in use and in environmental impacts. So I can't make a all-encompassing statement to answer that question.
Well, that was probably a really good summary of the field, and probably a good place to end it. We're over time. I just want to thank you, Paul, and I would suggest the next time you come, I really thought it was cool the way you personalized that graph of aquaculture production and key points in your life. So I think you should put another mark down when you gave this lecture, and then next time show us the update, put another mark down, and we can see how things are progressing with you and all your great work. So I want to thank everybody for coming and remind everybody about in 2 weeks Esther Guzman's talk. And I will do a clapping for all of us for Paul. And we hope to see you all in 2 weeks for Esther's talk. Thank you so much.